Can you remember? Can anyone of you please? Or Prabhudev, do you hear me?
Yes, sir.
You just tell me last day I discussed up to what?
Sir, last day up to binary fission was discussed.
Up to
Binary fission.
binary fission. Okay. So today bacterial reproduction I discussed binary fission. And after that, there comes to the conidia. In filamentous algae, what happens? From at the tip of the filament, some spore-like structures are formed exogenously in chain. The type of asexual spores which are produced at the tip of the filamentous filament exogenously, mean outwardly, in chain called conidia. And each one is conidium. So each one gets separated and give rise to new organism, new bacterium. So what are conidia? Conidia are asexual spores which are produced at the tip of the filament exogenously in chain. Then it is called conidia. After conidia, then comes to the endospore. In some bacterium like Azotobacter, what happens during unfavorable condition, a number of wall layers is formed around the protoplasm, forming endospore. They feel like they are telling you, endospores, I am drawing here. This is the protoplasm, the nucleate, around which you will get the wall, first wall layer, then you will get the so walls are pore wall, cortex, spore coat, and lastly Angiospermium. The 4 layers structure, and this is called endospore. So from inside to outside, endospore is protected by 4 wall layers. From inside to outside, the layers are the core wall, then the cortex, then the spore coat, and lastly the angiospermium. And in this period, they can actually stay for a number of months, even years. And during this period of time, it can tolerate even a very high temperature. And this is due to the secretion of from the bacterium. One substance is secreted called dipichlorinic acid, and this dipichlorinic acid protect bacteria from excessive heat. They provide heat resistance. This is what about the endospore. When favorable condition comes, under favorable condition, it germinates. We could call it germination of endospore. Under favorable condition, it germinates to give rise to new bacterium. This is what about the endospore. Example, Bacillus anthracis, to
Sir,
Clostridium. Wait. Tetani. I have to complete it first, then I will be asking you the questions, and you can also me the questions. So you see. So an endospore is formed in some bacteria so that it provides maximum resistance and it can protect the bacterial cell from desiccation. When unfavorable condition starts, in some bacteria, a number of wall layers are produced around the protoplasm, forming endospore. Already I discussed the wall layers from inside to outside are the core wall, the cortex, the spore coat, and lastly the angiospermium. When favorable condition comes, it undergoes germination to produce new bacteria. During this period of time, whenever it is in endospore form, from it one compound is secreted called dipichlorinic acid. It provides heat resistance. So this is what about the endospore. You need to remember 2 names. Those who can produce endospore: one, Bacillus anthracis, and Clostridium tetani. Now, what you are asking?
sir, can you please explain me the conidia again?
Actually, a lot of students are actually, only Gaurav Das, you unmute, plus Morgan and Abhay Das, you actually mute your audio. If you are asking question at a time, no one will be here. Now, who was asking now? Anyone?
Sir, me, Abhay Das.
Now tell me. A bit louder.
Can you please explain me the conidia again?
Conidia.
Yes, sir.
Okay. Now, next question coming from who asking another question? Who was asking again? Please ask now. Somojit Das ask. What a question?
Sir, what is endospore?
Endospore is an asexual spore produced under unfavorable condition in certain bacteria where the protoplasm is found to be protected by a number of layers and already discussed what are these layers. As they are produced inside, as the term comes, endospore. Okay, Somojit.
Okay, sir. Thank you, sir.
Next, any more question? Any more question from anyone? While I am discussing that, it is not good to ask me question because already it is the rhythm would be actually cannot be maintained. That's why I am asking you now. If you have any question, tell me now, please.
Sir,
Yes.
sir, what is the name of the last layer of that endospore?
Exosporium. 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 Anyone else? Any question? Okay. Now in conidia, what I said, conidia, actually all are asexual spores. Common thing about them is everyone is asexual spores. In case of conidia, what happens? The spores are developed exogenously in chain. Exogenously means towards outside. What happens? If it is a filamentous algae, suppose there is a filamentous algae, then for the tip of the filament, spores are produced exogenously. So as they are produced exogenously towards the outside, not inside, and they are found to be in chain. Three things: they are to be produced at the tip of the filament, point 1. Point 2, they are to be produced exogenously out towards the outside, and they are to be found in chain. If these three characteristics are satisfied, then you can call it you can call them conidia. And after whenever each conidium gets detached, give rise to new organism. So first of all, it is to produce the tip of the filament, then they are to be produced exogenously towards the outside, and then they are to be found in chain. And then only you can call them conidia. Who asked me the question? Avoid us. Is it clear now?
Yes, sir. Yes, sir.
Okay. Now we are proceeding towards the sexual reproduction type now.
ওর মানে ওই আর কি ছিদ্র
কিটা এন্ডোস্পোরটা
এন্ডোস্পোর একটা ছিদ্র আর কি
কিটা ছিদ্র এন্ডোস্পোর কাদা
অন্তরে
ডিপি কোলিনিক অ্যাসিড কিটা
ডিপি কোলিনি
Sexual reproduction.
গাদা গাদা পরে চালান আছে আইয়া কো
In sexual reproduction,
কোনিডিয়া কিটা কোনিডিয়া
আর এটি তো সব বাইরে
Type
ব্যাকটেরিয়া রিপ্রোডাকশনের নাম এটি তো আমি করতে পারতাম না
কইতে আমার এক্সডোস
conjugation,
স্পোরিয়াম এক্সোস্পোরিয়াম কিটা
অন্তরে
transformation,
স্যার আমার এটা
transduction. Conjugation, transformation, and transduction are the three types. In conjugation, what happens? These are two types again. Sterile male method, fertile male method. In sterile male method, what happens? Suppose there are two bacteria. The nucleate, and in one bacterium a circular structure is found which is called plasmid. So the bacterium, one bacterium plasmid is present, but in the other bacterium plasmid is not found. So it is called A plus or donor or male bacteria, or else it is called A minus or recipient or female bacteria. So here plasmid acts as fertility factor. So what happens next? A conjugating tube is formed between these two bacteria. Remember this thing. In this type of reproduction, the nucleate that carries vital genes not going to take part. So which one is take part? The plasmid. What happens here? One enzyme is going to cut it, and then so that it then proceeds towards the recipient or female bacterium. Then what happens? After that, in the meantime, plasmid enters the recipient cell. But you know, transfer is not complete because actually 90-minute time required whenever the transfer is to be started. Actually the process started with the formation of conjugating tube, but not to form conjugating tube, almost 8-minute time lost. And almost 8-minute time lost here, so complete 90 minutes will not be available for the transfer. So that when 90 minutes is over, automatically the process stops and eventually partial transfer occurs. What happens? Partial transfer. So that eventually so that what happens eventually they are called the. So what happens as a consequence of which the bacterium which was earlier F minus is converted to F plus. Okay, so this is what happened in case of sterile male method. I am repeating again. In sterile male method, here partial transfer of plasmid DNA occurs from donor to the recipient cell. Here two bacteria are taking part, one with plasmid, other without plasmid. The bacterium with plasmid called A plus or donor or male bacterium with without plasmid called F minus, recipient or female bacterium. Then what happens next? From the actually donor, the plasmid DNA is cut by endonuclease enzyme and gradually it proceeds towards the what? Recipient cell. And after being carried there, as 90 minutes time is not found for when transfer start and it ends, so when 90 minutes time is not available, that's why what will happen? Partial transfer takes place. As a consequence, a part of plasmid DNA is actually transferred to the recipient cell, and as a consequence of which the male bacterium remains male, and those who are earlier female bacterium, after getting the plasmid DNA, it would become male bacterium now, or both are now F plus. So what happens? Due to the partial transfer of plasmid DNA only from donor to the recipient cell, the recipient bacterium is converted to F plus or male bacterium, and in this way reversibility of sex occurs. And due to this reversibility of sex, the term used here is sexduction. The term used is sexduction. This is what about the sterile male method. Do you have any doubt? You can ask me now. If you have any doubt, you can ask me now. Okay? So we have to start with the hello. আমি ক্লাস করতেছি হ্যাঁ 10:30 পর Okay, so then comes to the fertile male. Is it clear now to everybody? Any question from anyone? It is clear that what is reversibility of sex? Hope so. Now I have to rub it out and start the fertile male method. Okay, note it down. Hope so. Is it okay now? Anyone please raise your hand and tell me. Soumyajit Das, repeat why there is partial transfer. Okay, I am telling you, listen. Another one, Sayatri Chaudhary, function of the acid, dipichlorinic acid, I think. Okay. Look one thing, whenever conjugation starts and it ends, if complete transfer of nucleic acid is required, then 90 minutes time is required for this. How much? 90 minutes. What happens here? Although during actually it is to time started from the formation of conjugating tube. So from till conjugating tube is formed, the plasmid DNA cannot be cut. It can be cut only after the formation of conjugating tube. So conjugating tube is formed. In the meantime, almost 8 minute times lost. So ultimately, for transfer we are getting 82 minutes. So 90 minutes is not available, and hence partial transfer occurs. Because from the time of conjugating tube, when 90 minutes is over, the process stops. Conjugating tube degenerates, so that further transfer is not possible, and that's why partial transfer occurs. Soumyajit, do you hear me?
Yes, sir.
Is it clear to you?
Yes, sir. It is now clear.
Now next comes Sayatri Chaudhary. You are seeing the acid. The name of the acid in endospore dipicholinic acid. This is actually it's a pyridine. Dipicholinic acid is actually a pyridine with two carboxylic group. This is dipicholinic acid. Pyridine with two carboxylic group. So DEEP provides heat resistance to bacteria, or it can protect bacteria from excessive heat. This is the role of the dipicholinic acid. Is it clear, Sayantri Chaudhary? Do you
Yes,
hear
sir.
me?
Yes, sir.
Okay, now I am to start the.
Sir, I had a question.
Tell me.
Sir, if both of the organisms become male, then how does a new organ?
One minute. Oh, just a minute, please. Now tell me.
Sir, if both of the organisms are if both of the organisms become male, then how does a new organism is formed?
No, actually conjugation is not going to take place between two organisms, two bacteria with fertility factors. It always takes place between one organism with fertility factor and the other organism without fertility factor. Getting me?
No, sir. Then how does a new organism? Then how is a new organism formed?
Then no problem. After this is formed, then it undergoes sexual reproduction to increase its number.
Oh, okay.
Getting me?
Yes, sir. Yes, sir.
So I am to start with the fertile male method. What happens here? Again I am telling you, two bacterium. This is the nuclear material. Wait a minute, please. Is it working? No. Just a minute. Look. So, now it is A plus bacterium. This is the nucleate of this bacterium, and this is also the nucleate of this bacterium. This is the plasmid. Then what happens? Similarly, then what first one thing happens here, you see. This is the nucleate, and plasmid is found to be attached with it. After this cell is called.
Sir, I have a problem in.
Who is asking?
Sir, me, Rohan sir.
I'll be giving you the time, not disturbing while I am teaching. Okay. After completing this, I'll be asking you the question. I'll be giving you time, then you will ask. Okay. In between, don't ask, please. So what happens here? You see, this is the plasmid. So here, what happens here? HFR cell is formed. What is HFR? HFR means high frequency of recombination. That means the recombination frequency of HFR cell is 1,000 times more than A plus cell. That's why term used recombination frequency. And the plasmid, then get attached to the nucleate, the plasmid nucleate is called episome. The plasmid that gets attached to nucleate is called episome. So the whole structure is not the episome. Only the plasmid is called episome. Then what happens? After that, conjugating tube is formed. Then what happens? After this diagram, then what happens? Right. You see, the nuclear DNA is cut here and it starts to proceed towards the recipient cell. After that, you see where is cut? The cut point is here. And again, same thing happens. Means, if I am going to draw a diagram over here now, after that, again partial transfer occurs. Region remains same. Just a minute, please. plasmid gets detached. Now it become A plus cell again. So here, fertility vector is not going to be transferred. I'll be sending you the ID and password soon. Anytime it would be stopped. So, what happens here? Okay. So ultimately, here in this case, the plasmid DNA is not going to be transferred to the recipient cell, so that even after conjugation, F minus remain F minus. Now, someone trying to ask me a question, tell me the question. What is the question? Tell me. Now you can tell me the question that you have, that you raised in your mind, please. Quick. Yes, Somojit Das, tell me. Unmute
Sir,
and
I can't.
answer.
Sir, I
Huh?
can't understand. I can't understand this part.
Which one? Which
Sir,
one?
sir,
Tell me.
total this part. Sir,
Total.
after the sir,
Huh?
when you have unmuted all, then you have started explaining these things, and I can't explain. I can understand these things.
Unmute all means?
Sir, I
What are you
mean,
telling?
sir, means when I have asked
Don't you hear the
another question.
class? You
Sir,
are asking
when
another question, then I'll tell you that while I am discussing, then you just wait for some time. Now I'm asking you to ask the question. What is the question?
sir, I can understand this part. This
Which one?
diagram. These diagrams.
Which diagram? This one? This one? This one? This one or this one? Which one?
Sir, total this part.
How can
I
it
can.
be possible? Already I discussed the sterile male method.
Yes, sir.
Already I discussed the sterile male method.
Sir,
Is
I
it
can
understood?
understand.
Sterile male method?
Yes, sir. I can understand this
Then what
part,
is the
but
problem here? Here I am repeating it again. This is the nucleate. Now it's a F plus bacterium. Now plasmid is found to be attached to the nucleate here.
okay,
After this attachment, the cell is said to be HFR cell or high frequency, the cell with high frequency of recombination. And this recombination frequency is almost 1000 times more than the F plus cell. It forms the conjugating tube quickly so that the degree of recombination is more. And then the plasmid then attaches to the nucleate called episome. What is episome? The plasmid then attaches to the nucleate, not the whole structure. After that, conjugating tube is formed. Now, in the nucleate DNA, an enzyme comes and creates a cut there. After that, the nucleate DNA starts to proceed to the recipient cell through the conjugating tube. Again, when 90 minutes is over, then the process stops. So that again partial transfer occurs. Now this partially transferred nucleate combines with the nucleate of the recipient cell, and then sometimes it is called merozygote. Because merozygote, why it is called? Because here union takes place between the complete nucleate of one organism and the partially transferred nucleate of the other organism. That's why the term used is merozygote. Then it can give rise to a lot of new bacterium by asexual reproduction. Now tell me, where you will not actually, what is the problem actually? Tell me please.
sir, I
Somojit.
understood now. Sir, I understood now.
Huh?
I understood
Okay.
now.
You understand? Okay. You understand now. Okay. Now, anyone, do you have any question? Okay. Now I am going to rub it out. Yes.
What is merozygote?
Actually, marrow means partial. You can call it partial zygotose. You can call it partial zygote also. This is because this zygote is formed. This zygote is formed due to the union of one complete nucleate and other partially transferred nucleate. As nucleate is partial, hence the term used is merozygote, or sometimes it is called partial zygote. Is it clear, Prabha?
Yes, sir.
Okay. Now I think it is completed. Then we have to proceed towards the transformation. In transformation, look here. This is one bacterium, and in this bacterium there is a capsule outside. So you can call it capsule. So this is the cell wall. So you can call it capsulated bacterium. Then heat so that the bacterium is killed, but the nuclear material remains intact. This is the other bacterium, and it is non-capsulated. Nucleate non-capsulated bacterium. After that, what happens? Nuclei of wall of non-capsulated bacteria. Then what happens? Nucleate of heat kill bacterium. nuclear of non-capsulated bacterium. Then what happens? Union of two nuclear occurs and eventually capsule is developed. Now it becomes a capsulated bacterium. That means by absorbing the nuclear material of a dead capsulated bacterium, a non-capsulated bacterium gets transformed into capsulated bacterium. Then by burning it, by binary, it can increase its number. The main thing here is that here what happens actually, the capsulated bacterium, actually the nuclear of the capsulated bacterium carry genes for the synthesis of capsule. And after being carried to the actually recipient bacterium, which is non-capsulated, these genes remains active. So that once union occurs, now each gene starts functioning and hence capsule is formed. That's why it is called actually transformation. Griffith performed an experiment by taking two different strains. One is virulent strain, other is non-virulent strain of Diplococcus pneumoniae. What happens here? You know, Diplococcus pneumoniae or sometimes Streptococcus pneumoniae, they can cause the disease pneumonia. So what he did? First, he injected some amount of non-virulent strain into some mice, and whatever they died. Whenever virulent strain is injected, what happens? The mice died. But whenever non-virulent strain injected to the same, then they remain alive. Then he killed this virulent strain. The virulent strain of the bacterium is heat killed and then injected. And it is found that the mice remain alive. Then he did one thing. The non-virulent strain heat killed virulent strain plus living non-virulent strain. You see, you get a mixture of these two, containing heat kill virulent strain and living non-virulent strain, injected to mice. And then what does it mean? You see, same thing, transformation occurs here. By observing the nuclear material of heat-killed virulent strain, which one is earlier non-virulent becomes virulent now, so that the mixture can kill the mice. But alone, if you apply or inject the heat-killed virulent strain alone, then it cannot actually kill the mice. But whenever it is mixed, due to transformation, non-virulent strain gets converted to virulent, and now easily the mice are died. So this is the basic thing in transformation. Are you getting me? Any problem? Do you have any problem, please?
Sir,
Yes.
sir, the last part of capsulated bacterium, how it forms? Please repeat.
This one.
Yes, sir.
What I said, when these two nuclear material gets fused, as you know, the nucleate of heat-kill bacterium possesses its own genes. Some genes are absolutely for the formation of capsule. As these genes remain active, so they start producing enzymes from these genes, and so then ultimately capsule is developed. You know,
Okay, sir.
the formation of capsule is controlled by some gene sequence, and genes are present where? In the DNA. Although it is heat killed, DNA remains intact, so genes remain functional. Whenever transferred to the non-capsulated bacterium, then what happens? Then these genes remain functional, start producing its enzymes so that eventually capsule can be formed. Getting me now?
Yes, sir.
Okay. So this is about the transformation. Last one is transduction. In transduction, what happens? This is the bacterium. Nucleate. This is the bacteriophage. Now, this first DNA inactivates this one so that the host DNA remains inactivated. And now cellular machinery is under the control of fast DNA, and he starts producing his component, different components. Eventually, host DNA. Now in the meantime, the fast DNA, by utilizing the cellular machinery of the bacterial cell, starts producing its own components like these. What are these? You can call them viral components. After that, assembly occurs and daughter phases are formed. After that, lysis occurs and the phase comes out. Then it is going to attack, and while coming out, if somehow the nuclear material of the host DNA gets attached to the spike part, then whenever it is going to attack a new bacterium, suppose it's a new bacterium, host nucleate, bacterial cell. Plasmid binds to it. Then ostein enters along with the bacterial DNA. After that, then the problem comes. Then if the union is required to take place, then what is required? So this is the host DNA, and fast DNA is required to be attached to it along with the bacterial genome. Remember this thing, a phage, whenever it enters the host cell, it do it like this, or it can be attached to the host genome. Whenever the phage DNA gets attached to the host genome, then the host genome will not be affected and even the host cell will not be affected. So at this stage, the phage is called, at this stage, you can call this phage genome as prophage, inactive phage. At this stage, phage is not going to kill or attack the bacterial cell. So if the union of two different bacterial genome is required to occur, then the phage genome is required to be present in prophage stage. If phage genome is present in prophage stage, then only union will be possible. So you are getting like this. And obviously phasm is also there. So this type of reproduction is called transduction. And till first genome is found to be attached to the host genome, the host cell would not be affected by it. That is, it is inactive stage. So in this way, this type of reproduction occurs with the help of what? The phage virus or bacteriophage. You know, bacteriophage is a virus that can attack bacteria. The viruses which attack bacteria are called bacteriophage, and such viruses, whenever they attack by lysosome, they create hole in the cell wall, and through this hole, nuclear material inside the enters the host cell. Then it can do two things. One, it is found to inactivate the host cell directly, or starts producing its own component, or it can be attached to the host cell. And if it is so, then whenever bacterial cell divides by binary fission, after every division, new bacterial cell will be formed. This is what called transduction. Now tell me, do you have any problem regarding bacterial reproduction, conjugation, transformation, and transduction?
Sir,
Yes, Ishita Sinha, tell me.
sir, what is mean by prophase stage?
Look one thing. A bacteriophage only within two stages. One is prophase, other is temperate phase. So whenever any bacterial cell, whenever any phage virus, after entering into the bacterial cell, binds to the host genome, and when bacterial cell divides, it is true, and it is found in the all progeny bacteria. In this case, the virus is found to be in inactive stage, and it is not going to do any harm to the bacterial cell. But till it is found to be connected, to the host genome. But as soon as, due to altered environmental condition or some toxic chemicals come in contact with this toxic chemical, then obviously this genome gets separated. Once it's separated, it enters the lytic phase or temperate phase stage. So whenever it enters the temperate phase, it is found to attack the host cell directly. In this case, what happens? It stimulates this host genome so that it can produce an enzyme, so that which in turn inactivate it. After that, cellular machinery is under the control of the phage genome, and it starts producing its own components. or sometimes you can call it virulent furrows. Same thing. There are two. This is the virulent furrows which can attack directly. Is it okay now?
Yes,
Who
sir.
asked me? Okay. So this is about the bacterial reproduction. Now, we have to proceed towards another thing is left. Yes, sir. This simply I am discussing cyanobacteria. Anabina nostoc. In cyanobacterial cell, actually few things I am telling you regarding, first of all, most important thing, the photosynthetic pigments. Most important thing is there, at first in prokaryote, photosynthetic pigments similar to hair plants is found. This is chlorophyll A is found. Then you will get other pigments, phycocyanin. The blue pigment, red pigment. So you see, in cyanobacteria, we are getting first time the chlorophyll A. That means not only in the eukaryotes, eukaryotic autotroph, but also in the prokaryotic autotroph also, chlorophyll A is found. That's why considered as universal photosynthetic pigment. In all eukaryotic autotroph, in all means all, in all eukaryotic autotroph and in cyanobacteria. Chlorophyll A is found. In all eukaryotic autotroph and in cyanobacteria, chlorophyll A is found. That's why actually it is considered as universal. Which one? Chlorophyll A is considered as universal photosynthetic pigment. Now, phycocyanin blue and phycoerythrin red. Remember this thing. Sometimes you know some Red Sea. I think you have heard about the Red Sea. The color of Red Sea is due to a cyanobacterium. Due to a cyanobacterium possessing this specific particular pigment. And this is the basic reason why it is red in color. So, and that is this cyanobacterium, you know, which is responsible for this is called trichodesmium. Then another most important structure I am to tell you, First of all, after photosynthetic pigment, second one is most important thing, heterocyst. You know, in case of bacteria, I will discuss this thing in the, suppose, rhizobium. Rhizobium is found in the root nodule of symbiotic bacteria, leguminous, root nodules of leguminous plants, and which in turn will help in the fixation of nitrogen. You know it, I think. If you go to the root nodules of leguminous plant, you will get some nodules over here. And these nodules are containing this rhizobia. And beside this, suppose here two things present. One, inside this nodule, you will get the bacterial rhizobium. Another thing present called lag hemoglobin. Lag hemoglobin is a chromoprotein synthesized by the leguminous plants, which is to act as oxygen scavenger. Act as oxygen scavenger so that the inside the nodule an oxygenic environment can be developed, which is very much required for the fixation of nitrogen. So lag hemoglobin is found in case of leguminous plant